for empathy and compassion. Of course, the Sanford Institute has really helped us put this together and has supported us in and out for these initiatives. And I also have to do a shameless plug for our symposium on empathy and compassion on February 2nd. So please go to the Institute's website, just Google the top the Institute website and we'll be there. So without any further ado, let me introduce old friend and colleague, Dr. Sarah Lazar. Sarah is really, I think, one of the first pioneers of the neuroscience of mindfulness. Sarah, did you publish the first brain imaging study on mindfulness?
Basically, yeah.
Basically, that was humbleness for yes. And
it was one pet study, but it was, you know, no one ever cites it. It wasn't very good.
Tiny sample size. Yes. So she has made incredible line of discoveries and has dramatically pushed our field forward, as you're about to see. And she's still cracking. I mean, now, I mean, you're really just doing an incredible job. She has over 15,600 citations. And that's only exponentially growing. She's an associate professor at Mass General and Harvard Medical School. And she has published some of the first work to show cortical thickness changes as a function of adaptive training, how mindfulness assuages affective, negative affect, and improves positive affect, and has just recently published a beautiful paper in Biological Psychiatry with respect to mindfulness practices and fear in the brain. She is a mindfulness practitioner for over 30-something years. She's all over the news, and I really am just so stoked that she's here today to present some work for us. So without any further ado, please help me welcome Dr. Sarah Lazar. Thank you, Sarah. The floor is yours.
Well, thank you, Fadl, for inviting me. That's a very nice introduction. And I said, your center looks gorgeous. I'm bummed that I don't get to come out and see you all in person. You know, it would have been great. So.
This was a different background. I'm sorry, I was just in a different meeting. Let me fix that.
Well, okay. It's like psychedelics too. Okay.
Yes, we're doing that now too.
Yeah. Okay. We'll talk about that some other time. Yeah. So you'll have to invite me next year too, though, so I can come out and see you in person.
Can't wait.
Yeah, it'll be great. Okay, so I'm going to share my screen. And I'm going to start that. All right. So can you all see that? Okay.
Mm-hmm.
Great. Excellent. Okay. So, right. So as Fadl said, I've been doing this research now for about 20 years. So I'm going to show you a little bit of, across what we've been doing before, but also some more recent stuff with fear conditioning and pain and anxiety. And so, and then there should be plenty of time at the end for question and answer. But also I always like to have an interactive talk. So if there's, you know, a burning question, you know, you want to jump in, just go ahead and just jump in, and we can have a discussion, you know, partway through. Okay, so I assume most of you know what mindfulness is, but just in case, right? So mindfulness, the classic definition by John Kabat-Zinn, is purposely paying attention to experiences in the present moment in a non-judgmental way, right? And for me, and so again, so it's the mindful versus the mindful. And for me, one of the key components of this is the non-judging attitude, right? And so this is, in Buddhist terminology, equanimity, right? It's I think lots of times people, when they first learn about mindfulness, the non-judging, they get hung up on the non-judging, because there's this idea like, you know, that, you know, everything is Zen, everything's okay. And, you know, it's sort of this like passive, like we're just supposed to accept everything as it is. And we are, but not the way that's implied by those phrases. And so I'd like to show this slide, and because everything is really important, as you'll see later on in the talk, that yes, it's accepting things as they are, but it's the key component is that it's in this caring and open way. So it's not apathy, right? So it's not just like, oh yeah, this is how things are. You know, it's not, we're not just resigned to this is how things are. We're not apathetic. It's, you know, we care. And we're open to the, you know, the unpleasantness of it, and we're experiencing the unpleasantness of it, but we're doing this as non-reactive way. And this is really the key to mindfulness, is it's this equanimity. So we're not just, you know, blindly walking around the street saying, oh, the sun is shining, and oh, isn't that nice? It's, you know, it's being open to it, you know, even if it was raining or if it was not such a pleasant day, you just be open to how it is, but in this caring and open, yet non-reactive way. So that's really, for me, the key element of mindfulness that's often gets sort of brushed aside. And I just want to highlight that because, again, we're going to come back to this quite a bit during the talk. Okay. So mindfulness, so that's mindfulness. So I often get the question, so there's mindfulness, there's mindfulness meditation, there's mindfulness stress reduction. What's the difference? So mindfulness, as you saw with the guy and the dog walking down the street, you can be mindful anytime, anywhere, right? That's mindfulness. You can do mindfulness in a extended way for 40 minutes, and just like set aside some time and just practice mindfulness. That's mindfulness meditation. And then we got mindfulness-based stress reduction, which is the first manualized clinical program for mindfulness, which again, probably most of you know, so I'm not going to spend a lot of time on this. Basically, it's just an 8-week program, and you practice mindfulness every day for 40 minutes. And then again, a key thing is doing the mindfulness anytime during the day. So as you're walking down the street, you can be mindful. While you're washing the dishes, you can be mindful. And the idea is that you're trying to become mindful 24/7. There's many scientifically validated benefits, including, I always like to highlight depression, anxiety, pain, and insomnia, because those are the big four for psychiatry. And doesn't matter what you have clinically, almost all clinical, even like physical things, often have depression and or anxiety and or pain and or insomnia as a comorbidity. And it's really clear, it's becoming more and more clear about the mind-body interaction. So even if you have heart disease or cancer or whatever, and you have these other psychiatric conditions, that the mood impacts the clinical symptoms. And so if you can decrease the stress and decrease the depression, anxiety, and the pain and insomnia, you can often have secondary benefits on these health benefits. And so I think it's really important to just recognize that how powerful is this, one very simple technique is for so many different conditions, either directly or indirectly. All right, and so it also been shown to increase attention and curiosity, memory, flexible thinking, creativity. I'm not going to go, I just like to show those because, hey, meditation does so many great things. I'm not going to talk about them today. Maybe write me back next year, we can talk about those things. Right, so as Fadl said, so way back when I first started doing this research, the reason why I got into this research was because I started practicing meditation and yoga, and I realized it was having this profound impact on me. Like I knew something in my brain was changing. And so, because it's not just you feel good while you're meditating, it really spills over into the rest of your day. And so I hypothesized, okay, doing neuroplasticity, you're not just sitting there, you're not just relaxing, there's something else. And so can we show that there's changes in brain structure and function? And I'm not going to show you the first studies today, but so the first studies basically show there's cross-sectional differences between long-term meditators and controls. Everyone just was like, well, they were just different. So then we did this study where we took people who had never meditated before, and we put them through the MBSR program, and we scanned them before and after, and compared them to people who were just scanned 8 weeks apart, but who did not go through the program. And what we see is that compared to the controls, there are several brain regions where there was more gray matter after the 8 weeks than meditators compared to the controls. And what are these brain regions? We've got the PCC, the TPJ, and the hippocampus, right? So for those of you who don't know brain, I guess I should have asked this, Fadl. So are most people here neuroscientists, or how much non-neuroscientists?
Most people are not neuroscientists, but most people are, I think, pretty informed. So I think if you just give a brief synopsis on what these functionally mean, then it would be probably most appreciated.
Okay, cool. All right.
Non-reactively and caring, though, of course.
Exactly, of course. Yes. Yeah. No, because I forgot to ask, because I assumed everyone knows mindfulness, but I thought, well, just in case. So, okay, but yeah, because I don't want to spend time on things that you guys you already know. All right. So the posterior singlet, it is involved. It's the number one region that's associated with mind wandering. It's also related to self-related processes. So when you see something, you say, oh, that's really relevant to me, is your posterior singlet who's trying to figure that out. The TPJ, the temporal parietal junction, it's right here next to your ear. It's involved in perspective taking. It actually does a lot of different things. One thing it does is perspective taking. And so it plays a very important role in empathy and compassion, empathy. It also is involved in attention. We're going to talk a lot more about it a little bit later in the talk. And then the hippocampus is the main part of the brain involved in memory. And what's also very interesting and important is that posterior singlet is the main hub of the default mode network, which many of you have probably heard about. So there's this idea that if you're just lying in the scanner, not thinking about anything in particular, there's a series of regions that get activated when you're not thinking about anything in particular. You would think most people thought that, well, just lying in the scanner, well, it would depend on what you're thinking about. Like, if you're just lying there not doing anything particular, and that, well, my brain would be very different than your brain activity because, we're very different people. But it turns out that pretty much everyone's brain activity in the scanner looks more or less the same. And the main region that's activated during that is the posterior cingulate. And then the TPJ and the hippocampus are two other nodes of the default mode network. And what's interesting is that, as I mentioned, that the PCC is involved in mind wandering, and it's actually, like I said, the whole default mode network. That's when you're mind wandering, not doing anything particular, that's the network that's on. So now, some of you might be thinking, well, hold on, it's bigger. And usually most people think, bigger is better, and bigger means more. And so does this mean more mind wandering? Well, so this is brain structure. And in the brain, we have both excitatory neurons and inhibitory neurons. And we think in this case, what's happening is that in the PCC, at least, that it's going to be more inhibitory neurons, because what's been shown is that this area gets turned off very strongly during meditation. And also it gets turned off, when you're focusing on something, and then comes back on when you're mind wandering. Also for people with Alzheimer's disease, the main regions impacted in Alzheimer's disease are the PCC and the hippocampus. Both of them get destroyed in Alzheimer's disease. And of course, that's associated with very great mind wandering. And so this idea is that these increase in thickness here, or actually density in these regions, is related to turning off the default mode network and regulating it more effectively. And we did not look at mind wandering in this study, unfortunately, but other people have shown this. The other thing we found was that there's a region in the brainstem that correlated with changes in well-being. So we gave people a questionnaire, a RIFS questionnaire on psychological being. So like, how happy are you and how satisfied are you with different aspects of your life? And so to ask about your work, your family, your income, your neighborhood. There's 16 different categories. And we found this increased pre to post, and that correlated with this brain region. Now, this brain region is approximately the area of the brain where dopamine, serotonin, and norepinephrine are produced. Again, we didn't assess those molecules in the study, but we hypothesized that perhaps that this change here is somehow altering those molecules. There have been other studies with PET imaging demonstrating that serotonin and dopamine are both impacted by meditation practice. And also, just theoretically, because often long-term meditators, it definitely has impacts on wakefulness and sleep, and arousal and whatnot. So it makes sense that particularly serotonin is likely impacted by meditation. So potentially this is related to that. We don't know. Again, some hand waving, but it's interesting to think about. Finally, what we found in this study was that the change in the amygdala correlates with changes in perceived stress, such that the more stress reduction people reported, the smaller the amygdala became. And this was interesting because it's been shown that the analogous part of the amygdala in animals does the exact opposite. So I couldn't find a good rat picture. It's actually done with rats, not mice, but I'm using Mickey, so sorry about that. So if you like Mickey. So what they did in the rat study was they had a rat at baseline, and they put it in its cage, or they had it in its cage, and then they put it in a maze. And normally if you put a rat in a maze, it will run through the maze and explore the maze, right? And so that was baseline. Then for 10 days, they put the rat in restraint, so they just a very small cage so it couldn't move. They just did it for a couple hours a day, but that really stresses rats out. And at the end of 10 days of doing this for a few hours a day, they measured the amygdala, and they found that same part of the amygdala that we found getting smaller got bigger. And also when you put the rat in the middle of the maze, it just sits in the middle of the maze, which is considered a stress behavior, because normally, like I said, it will explore the maze, but when it's feeling stressed out, it just sits in the middle of the maze and cowers. What was interesting is then they put it back in its home cage and left it alone for 3 weeks. And at the end of 3 weeks, they measured the amygdala again, and the amygdala was still enlarged. And when you put it in the middle of the maze, it still sits there in the middle of the maze. Now, like I said, this is the exact opposite of what we're seeing with, I don't know what happened to the person, with the humans, because with the humans, nothing about their home environment was changing. In fact, this was done back in 2008 when we had the the stock market collapse and everyone was losing their jobs and things were going bad. So life was pretty stressful at that time, and yet people were reporting having less stress and their amygdala was getting smaller. So in both cases, what we're seeing is it's not so much the stressor, but the reaction to the stressor that is what we're seeing here. And that's really important because, again, just because you start meditating, it's not like your life is just going to suddenly get better, right? You still have to commute, you still have to deal with deadlines, you still have to deal with all the crazy people in your family, right? Nothing about your environment's going to change. And what changes our relationship to those stressors. Okay. So I mentioned before that some of the most important. What's that?
Just to ask a quick question in the interest of the interactive presentation, I'm wondering, so are you saying that because you observed the opposite thing in rats, you're then taking that assumption onto humans? My understanding is that there's a correlation between more area in the brainstem and increased well-being. But in rats, when you perform this experiment, you don't see that.
You don't see okay, so one's the brainstem, one's the amygdala, so those are different. So in the brainstem, we just have humans and we just have self-report in humans.
Right.
In the amygdala, we have self-report in humans, and then we just have the rat data, and it's not our data, just showing basically that that brain region is related to the relationship with stress, right? So I don't know if that's your. So what exactly is your question? I guess.
Well, so I guess the part of asking this is the increased area, the increased what you see in the fMRI within the brainstem is correlated with more well-being, right? So the assumption there would be that there's a greater attention or these mindfulness, and that leads to this increased area in the brainstem, right? What about people who haven't practiced mindfulness but have an increased area in this brainstem? What is that? How does that correlate?
Well, I also want to just say it's important to say this. It's not fMRI.
It's structural MRI.
MRI.
Okay, structure. Okay.
Well, it's not blood cells. She's showing greater concentration. Is it concentration or thickness, density?
Concentration, yeah. So more, so it's dark. It's the neurons are more. There's more densely packed neurons. Probably, and so yeah. Sorry, I've been giving this talk to non-scientists a lot, so I sort of gloss over a lot of this. So I should have just gone back over this. So so we're looking at gray matter density.
James is a scientist.
All right. So okay. Well, yeah, but it's sort of because you guys know, and so you need the details, right? So we're looking at gray matter density. And so the idea is, so in MRI, structural MRI, all you can tell is how gray is that voxel. And the idea is that the more gray it is, there's more neurons in there. The more white it is, there's more, white matter or other things in there, in that voxel. And so it becomes more gray. So it suggests that the density of the dendritic spines and whatnot are more dense, become more dense. Right, that's the underlying cellular mechanisms, potentially, maybe, who knows, right? It could also be potentially blood vessels. And also, because blood vessels we know can change in diameter so like, again, nothing to do with humans. So in rats running through mazes, you can chop up their brains, so scan them and then chop up their brains. And what you see is that this increase in gray matter density in rats, sometimes it's because of increased synaptic branching. Sometimes it's because, again, of these bigger blood vessels. And it can also be from increases in astrocytes and glia and whatnot. And all three of those can contribute to changes in performance in animals. So in humans, we don't know. Right, what's going on. So again, when I glossed over with the rat study with the stressing them out for the 10 days and then putting them in the maze, in that case, they had chopped up their brains. And what they saw is that it was actually the dendritic spines that were growing. And that the increase in the dendritic spines was related to the stress and to the behavior. So they could actually see at the cellular level what was happening. And of course, we can't. Some days I would love to chop up my subject's brains, but we can't do that to them. Anyways, and so we don't know exactly what's happening at the cellular molecular level. I mean, obviously also because gray matter density is very, in terms of, is a pretty gross measure. And you think about it in terms of a very coarse measure. Obviously there's probably ultra things happening at the molecular level that we're not getting. And there could even be like small changes in dendritic spines and whatnot, and glia that we're just not picking up because it's just not sensitive enough. So that's at the sort of the molecular and cellular level of what's going on. And so, right, so going by analogy with the rat study, we're thinking that potentially it's a decrease in the branching within the amygdala. And that's what's related to the decrease in stress, because it is similar in that it's not, and it's the relationship to the stress as opposed to this reaction to the stress in real time.
Mm-hmm. In the human study, was that only people practicing mindfulness, or was it across the board?
No. And so that's interesting. So in that I actually combined two different studies there. So the amygdala study, we just had the meditation group. Since then, we haven't published this yet. We've done 2 more studies, and we've seen the same thing, that the change in stress correlates with the change in the amygdala size. And it's really clear it only happens with meditation. So in one study, which I'm going to show you in a little bit, the controls did exercise. And so they reduced their stress by exercising, no change to their amygdala. In the other study, they did like a health education control. We had 3 conditions. We had health education, we had mindfulness, and we had yoga. And both the mindfulness and the yoga, again, really effectively reduced the stress. But even yoga didn't change the size of the amygdala. Only the meditation changed the size of the amygdala, which is really, really cool. And so we're going to need to write that paper up. So really just show that it is something specific to meditation.
Got it. There's an interaction.
Yeah. Any other questions, as long as we're stopped? Nope. Cool. All right. So we'll carry on. Where are we? All right. So, So let's talk about pain. All right. So this is a study done a couple years ago by my student, Tim Gard. And so again, I know a lot of you know about pain, but it's just for those who don't. So basically, there's a lot of different ways to deal with pain. So when the class can distract you, right? So if the doctor will, before he gives you a shot, will say, "Hey, look over here," and while you're looking over there, they give you the shot. And if you're not paying attention, you don't notice the pain so much. You can modify the expectation. Say, "Oh, this is not going to hurt a bit, right? Or it's it's going to hurt a little bit, but you won't bother you too much." And of course, you can do placebo, right? Which is Fadl is, the master of the placebo, right? Give them a cream and say, "Oh, this is going to block the pain," and if they believe you, it does block the pain. And what's interesting is that in terms of the neuroscience of what happens in the brain, they're fairly similar. So here, oops, come back. So this is your brainstem. Well, here's your spinal cord up into your brainstem, right? So the noxious input comes in, goes up into your thalamus, and then into sensory cortex, the cingulate, the insula. And so the blue network is sort of the sensory network. And then this brown network is the front of the brain, and that's sort of the executive control part of the brain. And what we see is that for all 3 of these forms of pain control, what happens is that the front of the brain turns on and turns off the blue network, and basically tells your brain, just don't feel it. So the signal comes in and it comes up, and then it just stops and it doesn't get processed. And so you don't feel pain. Like you literally don't feel the pain because your front of your brain says, just don't feel it. So you don't. So we wondered what would happen with meditation. And I'm going to say, so this is very importantly, in this study, we use long-term meditators, right? So these are people who practice Goenka meditation. For those of you who know about Goenka, it's just a form of Buddhist meditation. On average, they had 6,000 hours, which is quite a bit of lifetime practice. And they were matched to 17 controls, right? And just so, because some of you who know Fadl's work knows that he's done a lot with people with just like a week of practice. And so our results are a little bit different from Fadl's. So if you know that, and probably because of the week of practice versus 6,000 hours of practice. But I'm just gonna tell you about what we've found. So what we did is, we put them in the scanner, and first they went through and we told them to just lie there and you're gonna get zapped. There's a little buzzer on their hands, or like the thing like where you shake someone's hands and there's that thing there, and it's sort of that sort of shock. So it's more or less that's level of shock applied to the back of their hand. And so there was a 40-second period where they were shocked at some point during, weren't told when, they were just lying there and suddenly they would get shocked 3 times. And then they rated it on a scale of 1 to 10 about how intense how intense the sensation was, and also how unpleasant it was. And again, it's been well established that there's 2 components to pain. So one is the actual physical sensations of the pain. So the stabbing, stinging, throbbing, burning. And then the other part is the unpleasantness, the I don't like it, make it stop. Why is this happening to me? Right? And those are separate. And so they know behaviorally that if, you can give the exact same pain stimulus to 10 people, and more or less, they will say, yeah, on a scale of 1 to 10, it hurts about a 5 or 6 or whatever. But in terms of the unpleasantness, it can be all over the map. And that has to do with, how their past experience with pain, how they were taught to learn to deal with pain. So the 2 are separate, and they're separate in the brain, and they're also separate behaviorally. So we had them rate both the intensity of the pain and the unpleasantness of it. So first of all, we went through and they did it in just a normal state, and then we taught before the controls went in, we taught them how to be mindful. And so then we told them to do this mindfully, right? And so the question is, what happened in their brain?
Really quick, I think I remember this study, but were they always mindful first? Or was it counterbalanced or randomized?
Good question. I'm pretty sure.
Yeah, go ahead. Sorry.
I don't remember.
That's okay. It was a while ago.
Yeah, I should know this. You're the first one who's ever asked me that. I think it was counterbalanced because Tim would have done counterbalanced, but you never know.
So he's German, so you know.
Yeah, yes, must do by the book. All right, so what did he find? So he found, remember, for all other forms that we see is, go back, that the front of the brain turns on and the back of the sensory cortex gets turned off. So what he found was that basically the opposite. Go back. The front of the brain got turned off. So here's the mindfulness practitioners, here are the controls. For the mindfulness practitioners, the front of the brain got turned off, controls turned it on. And sensory cortex, insula in this case, mindfulness practitioners actually turned sensory cortex on and the controls turned it off. So now let's pause here for a moment. What's going on here? Right? So basically the exact opposite of what we see with placebo and everything else. So what's happening? So let's remember the instructions for mindfulness. Mindfulness is paying attention to the present moment, non-judgmentally, right? And so this is what we're seeing here, is that they're paying attention. They're really paying attention to that sensory cortex, right? So with placebo and distraction, distraction in particular, like you're told, don't pay attention to the pain, pay attention, to that thing over there. And so here they're like, no, actually pay attention to it, but do it in this open, accepting, non-judging, caring way. And that's exactly what we're seeing in their brains, is they're turning off the judging and they're turning on the sensory experience of it, right? And the controls, although they're trying to be mindful, they've only been able to do it for 5 minutes, they don't really have any practice with it. So they're basically doing it the same way, that everyone else tries to turn off pain. What was interesting is that when we looked at the self-reported ratings, what we saw is that basically there's no impact on the intensity, so the burning, stabbing, tingling sensation, but there was a 20% decrease in the unpleasantness, the I don't like it, make it stop. So even though they were feeling it more, they were okay with it. It was just like, okay, I'm getting zapped and it's stinging and stabbing and, burning, but it's okay. And that's what equanimity is all about. So there's my little equanimity balance. So this was pretty cool. Like once initially we were like sort of, what's going on? But then this was nice. And so I like this quote from Sylvia Boorstein, which is, life is painful, suffering is optional, right? And again, this gets back to this idea that.
Sarah? You said that's what equanimity is about. I was very interested in your saying that, talking about the equanimity piece. And so is this, using that first art, second, does this fit with the first art, second art paradigm? So that what you get is living at the place of the first art. In other words, okay, you still experience the intensity, but the unpleasant changes. Thank you.
Exactly, yes. Yeah, for the boost out there, right? This is the second dart. It's right. They've turned off the second dart. Exactly.
Thank you.
Yeah, and right. So the start sharing again. right. So life is painful, suffering is optional. Because again, you still have the deadlines, you still have all the stressors in your life. That is not going to go away, right? The pain is not going to go away, but the suffering, how you respond to all those things in your life, that is going to change. And that's really important. And so this is really, really what we're showing in a really profound way, that, at least with long-term meditators, that you really can see this change, even to physical pain. And then the whole brain reaction to it actually changes. All right, so now this again was long-term meditators versus controls. Sorry. This is the work of Josh Grant, who basically found something similar. So again, he found the front of the brain turning off, and the thalamus, which again is parasensory cortex, turning on. So same idea. The one reason I'm showing him is that he also did functional connectivity, and actually showing that the connectivity between the front of the brain and the sensory cortex decreased the more it became uncoupled with pain sensitivity. So the more they were saying that, the less pain intensity in their case that they reported, the less the front of the brain and the back of the brain were talking. So again, it's just sort of just turning off the control of the sensory cortex. And also that this was correlated with the years of meditation. So again, the more you practice, the more these two brain regions come uncorrelated, and the more you're just experiencing it and not trying to control it.
How long were the meditators trained again? Was this MSR?
In my study, it was 6,000 hours.
So that was a minimum. They had to be long-term meditators?
They had to be long-term meditators. That's the average. I don't remember. They had to have gone at least one retreat, I think. And these people had all, I think he recruited them from a Goenka
center. Not Josh's study, your study. His study comes from a Zen retreat center, and their minimum was dramatically large. But your study, was that MBSR or was that long-term trainer?
No, it's Goenka, because he did it in Germany. And so it was a Goenka center in Germany. And so they had all gone on at least one Goenka retreat. I don't know, I don't remember the details of how many.
Thank you, Sarah.
Yeah, they were long-term though. Yeah.
I'm so sorry for interrupting you. Thank you.
Yeah, no, that's totally fine. That's totally fine.
Hey, Sarah, let me ask you a quick question, if I could.
Yeah.
Decoupling of executive cortex and sensory cortex, I get that part. But decoupling is an active process. Right? I mean, one doesn't, there's in fact something going on in frontal cortex that is being dampened. And so my question is that the beginning of allowing one to be aware of something and not be reactive to it? Or is that some secondary? In other words, does A go to B or does B go to A here? What's the sequence of activation, do you think?
Well, we don't know.
Happens with mindfulness.
So, all right. So this is totally hand-waving speculation. My guess, because the idea is, the goal is you want to respond rather than react. And so I think the idea is that you're forming a completely different pathway, because ultimately you do have to make a decision on this data, right? And so it's not like, because again, it's not like, okay, I'm just going to sit there and let the person beat me up, or I'm just going to sit here and endure the pain, and I'm not going to, like if I burn my hand, I'm not just going to keep my hand on the fire, right? Or on the burning, I'm going to pick it up. But the idea is that, and move away from danger. And so, but the idea is that, there's a pause, and that it's a different way of processing the information. And so my guess is that it's not necessarily that you're dampening the front, is that that automatic connection's not there, right? Or that there's, I don't know. Again, this is me speculating wildly, right? So that there's just a different way of, probably a different pathway through which the information flows, is my guess.
Yeah, that was the concern about the word decoupling. I get it at one level, but there must be a coupling involved so that one can both feel the pain and decide to deal with it differently.
Right. So the thing is, he looked specifically in the regions that were different between the meditators and the controls. And so it could be that there's some other region somewhere else that he's not assessing that. Yeah. And so I just don't know.
Thanks much. Appreciate that.
Yeah, sure. All right. So, and we're doing good on time. We're fine on time, right? All right. So we're going to talk a little bit about fear and anxiety. So, and you'll see where I'm going with this. So, exposure therapy. What is exposure therapy? So, say you have some sort of phobia, and you go to see a psychotherapist or mental health professional. Say you're afraid of spiders, right? One way for dealing with phobias is something called exposure therapy. This is also how they deal with PTSD. And you have these memories that are traumatic, and with often what happens is that our brain can't deal with it, gets overwhelmed, and so it just shuts down, and you start, and that's why you start having maladaptive responses to the traumatic memories, and also why for people who have phobias, it's just like, okay, I just can't deal, like you just shut down. And so the idea of exposure therapy is that you gradually expose the person to whatever it is that is they're phobic to, or the traumatic memory, but you do it in small bits and pieces in a very controlled manner, so that gradually you become more used to it and you can deal with it, and you learn to have a more adaptive response to it. And that the more you're exposed to it, the more the fear goes down, right? And so there have been some studies with exposure therapy with PTSD showing that well, actually, I'm jumping ahead. All right, never mind. Okay. So now what is mindfulness, right? So mindfulness is paying attention to the present moment in this non-judging, open way. So we hypothesized that then mindfulness would be useful. And it's been shown that mindfulness is useful for phobias, but then also for all sorts of anxieties, so including PTSD and phobias and social anxiety, which is basically phobia of talking in front of people, and general anxiety, right? And so we hypothesized that mindfulness might be working in part by providing an optimal condition for exposure to aversive stimuli, right? So for the people with the phobias, if you can learn to be open and non-judging and non-reactive, but yet still caring, then, that's how you can, enhance your ability to allow yourself to be exposed to the phobogenic stimuli or the traumatic memories, and, heal more quickly. So that's one of our hypotheses, right? So to test this, what we did 2 experiments. I'm going to sort of show bits and pieces from both of these experiments, because one was a pilot, and then with a smaller number of people, and we did it again with a bigger number of people. And so I'm just going to show, and we did them slightly differently, but I'm just going to show the results intermixed. So the first one was done by my first postdoc, Britta Holzel. And then the second one was the one that Fadl mentioned that got published last year in Biological Psychiatry by my other postdoc, Gunesh Savinch. And so the first one was Britta. So in both of them, what we did was a 2-day fear conditioning protocol, and I'll explain that in a minute, both before and after MBSR. In the first study, they were randomized to mindfulness-based stress reduction or to a wait list. And it was a 1-to-1 randomization, and it was people seeking stress reduction. The second time we did it, and this was, I think 60 people total, so 30 and 30. The second time was they were randomized to MBSR or to exercise. And this is the one I mentioned before, where both of them reduced stress, but only the amygdala changed in one of them, only in the MBSR group, not in the exercise group. Also, which is important, is that everyone was told that there was no control group. They were told that both conditions were equally efficacious, and that we were just trying to see how they were similar and different in the brain. The exercise group also got information about diet and sleep and hygiene. And both groups are told to practice 40 minutes per day at home. And so we really tried to match them as much as possible for expectation, as well as for attention and home practice, and really tried to make them as equal as possible, so that we really could see, okay, what is mindfulness versus what is all the other effects of the program. Okay. And also both groups had a half-day retreat in the 6th week, so which is a classic component of mindfulness-based stress reduction, which is where you spend a day being mindful. And for the exercise class, they exercised a couple times, and we gave them some healthy food, and we talked about diet and massage and humor, and some other good stuff. All right, and so we did the same thing in terms of we put them in the scanner, we gave them a mild electric shock, but this time the mild electric shocks was paired with colored lights, right? So this is classic Pavlovian conditioning. See a light, get shocked. And there's 3 different colored lights. And the lights were always counterbalanced both across people and pre to post. And so 2 of the lights, they got shocked. I think it was a 30% ratio. So 30% of the time when they saw the blue or the red light, they got shocked. The yellow light, they were never shocked. That was during conditioning. Then immediately after that, one color was extinguished. So in the second run, they saw the blue light and the yellow light, but they didn't get shocked at all. And so here they learned that the blue light now means that it's safe, but they have no new information about the red light. We then bring it back 24 hours later, and we then just show them the lights, but we don't shock them. Also, I forgot to, come back. For those of you who are into fear conditioning, you can see it here. Oh, actually, no, you can't see it. So here, during the conditioning, this was the room, like the lights were here on this desk. And then, and that's these were represented by the Pentagon. But then during the extinction and recall phase, there was a different office that looked very, very different. And so, so for those of you, I'm not gonna go into it, but that's an important detail for those of you who know about fear conditioning. Anyway, so the extinction, they learned nothing new about the yellow and blue lights. And then they updated their information about the yellow and blue lights, but they learned nothing about the red light. And then on day 2, we said, okay, how are they responding? And in particular, the question is, how do they respond to the blue light compared to how they respond to the red light and the yellow light? Okay, how are we doing? We're doing good. Okay, so as I mentioned, exercise is a great way to reduce stress. Both groups reduce their stress very effectively, right? And that was the same in both groups. What was interesting is that for the emotion regulation and anxiety, that was their group difference. So the MBSR improved the emotion regulation and anxiety versus there's no change in the SME group. Home practice correlated with the changes in stress and emotion regulation. And so now in terms of fear learning, this is what we saw. So I'm going to talk you through it because it's a little bit. So for the controls, this is the skin conductance response. At baseline, at pre, so the red is the lights that they got shocked to versus the light that was, the yellow light that was not shocked. And this is during, I should say, this is during conditioning. This is not during day 2. This is still day 1. So day 1 while they're being shocked, right? So this is their skin conductance to being shocked at baseline. And this is it at post. And then here's the same thing for the meditators. So let's talk through the controls first. So we see this big reduction in the skin conductance response to being shocked. What's going on there, right? And this is actually well established that, people still, there's still a difference between the two, being shocked and not being shocked. So they're still responding, but it's not as big a difference as it was at baseline. And basically what's happening is that, this is 8 weeks apart, that they remember, right? They remember from 8 weeks ago that they're gonna get shocked. And like I said, we learn what pain, the classic way to control pain is for the front of your brain to say, just don't feel it. And what we're seeing here, we think, the way we're interpreting this is that basically this is what's happening from pre to post. They know they're gonna get shocked. And so they're trying not to feel it. They're trying to control it, and they're managing that, right? So they're having less of a reaction because it's so unpleasant. And so they're having less reaction. So they're still reacting, but it's nowhere near as big as it was at baseline. Oops, come back. In the MBSR group, what we're seeing is that there's no difference from pre to post, right? They're responding to that being shocked just the same much as they were at baseline. And again, at first when we saw this data, we're like, what's going on? But then we thought about it, right? This is the same thing that we saw before with the pain study. It's the equanimity. It's that they are, they're not being defensive. They're being open and allowing themselves to be shocked. And they're knowing that, okay, this is going to happen. There's nothing I can do about it. So I'm just going to let myself be shocked. And they're having the full reaction to it. And you could say, okay, well, this is just waving our hands. But what we found is that the stress reduction correlated. So the more similar they were at pre and post, the more stress reduction people reported. Versus the people here, there was less stress reduction. People who look more like this had less stress reduction. People who look more like this had more stress reduction, right? And that was really strongly correlated. And so it really does suggest that this ability to stay open and just allow yourself to be shocked, or just let things happen the way they are, that it really is adaptive, right? And that it's powerful, right? And so from the Buddhist perspective, this would be beginner's mind, right? Literally beginner's mind. They're responding just the way they did 8 weeks ago. In that first study, we also looked at white matter, and we also found that the this differential change in response to getting shocked correlated with changes in the uncinate fasciculus, which is the major white matter tract between the front of the brain and the amygdala. So again, it's suggesting that this, this change in how the amygdala and the front of the brain are talking to each other is related to, this ability to just be okay with things. All right, what about day 2? So again, now we're looking again at here and looking at to see how things have changed on recall. And so first we looked across the entire brain. And what we said was, okay, so just, how are meditators different than controls? And the main region we found was here, which is the supramarginal gyrus, which is basically more or less the same, it very largely overlaps with the region that I showed you back at the beginning of the talk, right? I showed you that there's 3 regions that grew in gray matter thickness or gray matter density from pre to post 8 weeks of practice, right? This supramarginal gyrus here. And it's basically the same region here. So this is brain structure. This is now brain function. We're showing that this area is now more active during recall in the meditators compared to the controls. Okay, and I'm going to talk more about this. I'm going to show you some data, then I'm going to talk through what it means. And actually, so what I'm going to say here, so before I told you that it's involved in empathy and compassion, but it's also involved in selecting what you pay attention to. So whether you're paying attention to me talking or whether you're paying attention to, what's going on inside your body, right? So external versus internal, and selecting what you're paying attention to. That's what the SMG does. And what we found is that the activity here correlated with practice time. So the more people practiced, the greater this activity was. So now the main reason why we did this whole study was because of the hippocampus, right? Because this fear conditioning was originally constructed as a learning paradigm, right? For years, it's considered the classic learning paradigm. But it's also, of course, a great emotional regulation paradigm as well. And so we're interested in the case of the hippocampus. We want to learn more about what was happening with the hippocampus. And so all of our hypotheses are actually about the hippocampus, because extinguished memories must be remembered. And we also know that individuals with anxiety disorders have difficulty updating the stimulus response association, right? So when you put people with anxiety disorders and do this in the scanner with them, on day 2, they don't remember that the light was extinguished, right? So let me go back here. Right, so people with anxiety disorders, so most people remember that the blue light was extinguished and that blue light means that they're safe. People with anxiety disorders forget this, and they tend to respond to the blue light the same way they respond to the red light. They forget that this has happened. And it's because of their hippocampus, and they've shown that the hippocampus, that the structure of the hippocampus predicts their ability to remember what happened on day 1. And we know the hippocampus is crucial for episodic memory. Right, so that's what I just said, that is anxiety disorders. And so what we saw is that, again, for just the skin conductance, that there was a difference between the 2 groups, right? So the meditators were better able to remember that the blue light meant that they were safe, and the red light, they had compared to the red light. And also we see this difference in hippocampal activity. So though I should take that back. Now these are the same. So they're basically the same. So both groups were changed, because again, exercise is very good. So the hippocampal activity itself wasn't different between the 2 groups. But what was different was then we looked at hippocampal connectivity. right? So activity was the same, but connectivity was different. And what we see here, this is the hand region of sensory cortex. So this is the area where they're actually had gotten shocked the day before, right? This is day 2, so no one's getting shocked on day 2. But while we're doing this, while they're lying in the scanner, they have more connectivity between the hippocampus and the hand area where they were shocked the day before. Furthermore, they have decreased activity in visual cortex. So let's pause about this, right? And we also have, as I mentioned, the change in this area, which is involved in selecting what you pay attention to, right? So putting these 3 together, like I said, this says, okay, you're paying attention external or internal. So this suggests that here we're having increased attention to sensory cortex and decreased, activity to visual cortex. And so the vision would be the paying attention to the lights and what's going on, in the scanner versus paying attention to what's actually happening in their body, right? So let's just pause about it and think about this through, right? So this idea is that then that they're in a condition where they know they've been shocked in the past, they don't know what's going to happen. And so rather than paying attention to the lights, they're paying attention to their actual sensory experience, right? That's what this suggests, right? And that they're choosing to pay attention to the sensory experience rather than to the information on the screen, because the information on the screen has been changing.
Can you show us that sensory area that you were referring to, Sarah?
Yeah. That's definitely the hand area. I showed it to sensory.
That's hand?
That's definitely hand, yeah.
Okay, so basically, so there's greater connectivity between the hand and the hippocampus, and from pre to post, or just at post by group?
Pre to post.
From
yeah,
pre
both.
to post, but not by group, correct?
CSC, MBSR only within group, yes.
Only within group. Okay.
Yes.
And then what you're showing in the next slide is that the inverse of that hippocampus seed is in fact showing stronger connectivity with V1.
Less decrease.
Less connectivity with V1.
Yeah.
Okay, and that you're inferencing that connectivity is them being more in the body, not the scanner?
Right. So, right. Because the visual would be the paying attention to the lights. So there's less information about the lights, and it's more about what's actually happening in the body.
Okay, and is this during the light, during the stimulus, or the entire scan?
This is day 2. So day 2, this is right. So what happens is they're seeing the lights continuously on day 2, but they're not getting shocked.
No. So I'm sorry. Let me be clear. The connectivity here, is this seed-based connectivity? Is this a PPI? Are you looking at the stimulus, at the light, or are you running connectivity from time 0 to the end of the scan? Because if it's during the whole scan, right? So it's different, because if it's during the light and there's more V1, that makes more sense.
I don't know. I'd have to go back and ask her. I would assume it would be during. What do you mean by during the light versus during the
so the presentation of the light before the stimulation?
Because they're getting seeing the light the whole time, though. The because it keeps the chain. They can see it like 50 or 60 times. So they'll see red light, green light, red light, blue light, blue light, blue light, blue light.
I'm sorry. Let's back up. They're getting stimulated during the light presentation?
On day 1.
Okay.
So on day 1, hold on.
I think I understand, but then after how many trials do they just realize that they're not going to get pain anymore?
We're about to get to that.
Sorry, please continue.
Right. So day 1, so the thing is they only get shocked in the very first scan on day 1, right? So this is like a 10-minute scan. So they're getting shocked intermittently during this scan. Then they have another 10-minute scan where they only see the yellow and the blue lights and there are no shocks. Then on day 2, no shocks, 10-minute scan, they're seeing the lights the whole time.
But there's no shocks in day 2.
No shocks in day 2. It's all just recall. Okay, so now, right, so that's a very important question. So what's been shown is that people actually learn this pretty quickly, right? And so other people show that so not in the scanner, but outside the scanner, but basically people learn, within 2 or 3 shocks, that they're not going to get shocked. Right? Then they see the blue light a couple times, they're not getting shocked. They're like, okay, I'm not getting shocked this time.
And they have the thing attached to them.
Yeah.
They have the probe attached to them. So
right. And we tell them you may get shocked.
Okay.
Yeah. So yeah. And so, but again, this is at post though, because if they remember, it depends on how much they remember from before, right? Because because there's day 1, day 2 at baseline, there's day 1, day 2 at post, right? And so how well do they remember from the post that baseline they weren't shocked on day 2? We don't know. But okay, so on day 2, so the first just 2 or 3 pictures presentations, they've shown, many other people have shown that exactly what you're saying, that there's certain brain activity involved in the initial selection and figuring things out, and then there's in the maintenance, right? So in the early selection of which memory will to call, and then the ongoing learning, because again, so it's like, okay, well, blue light, okay, I wasn't shocked, but you never know, maybe I will get shocked, right? And so they're ongoing learning the whole time. And they've shown that these are different and you can separate them and whatnot. And so these are separate functions. And so we also looked, so what I just showed you was the whole 10-minute scan. So now we look at just the first, I think it was like 20 or 30 seconds of the scan. And what we saw was this. And so this, what we found, again, this is the meditators versus controls, pre versus post, that and this is a functional connectivity, right? With the hippocampus, was the Dlpfc and the Retrospinal Cortex, right? And so Dlpfc is important for suppressing the old memory, the incorrect memories, right? So if initially you learn that the red light means you're gonna get shocked, then you remember that, well, I take that back. So first you learn that the blue light means you're gonna get shocked, and then you learn that the blue light means you're safe. So then when you see blue light, you need to repress the initial memory saying blue light is shock. And you have to correctly remember that the blue light means you're safe. So the Dlpfc is involved in suppressing the first memory that says, hey, you were shocked. And then, oops, come back. The retrosplenal cortex is about context. So again, I mentioned that there's 2 different offices in which they were shocked, right? And so this is where the retrosplenal cortex is coming in, because it's saying, okay, blue light, but also blue light in office 2 versus blue light in office 1. So this is how they're remembering that, okay, yeah, blue light, so it's blue light, but it's also office 2. So I know, so that's the context dependent learning. That's the retrosplenium. So both of these regions are coming on here during the early selection. And again, this is selectively in the meditators. And then the other regions are coming on throughout. So suggest is sort of as what Flettel was just suggesting is that, so it's both, the mindfulness is helping both with the initial selection of the correct memory and the suppression of the incorrect memory, and in the continuous updating of information to say, hey, yes, right now I am safe and I'm not being shocked. And so I can just be in my body and just relax, and that's okay. So, so basically from both of these studies, the main conclusion is that mindfulness of the body is key for both pain and fear coping, and that we're shifting from thinking about and defending against threats to experiencing it openly and non-judgmentally, right? And so a couple of quotes, of course, FDR. So the only thing we have to fear is fear itself, nameless, unreasoning, unjustified terror, which paralyzes needed efforts, right? Because again, this gets to this idea of responding versus reacting, right? So if we have the fear and we're like, oh my God, you can't respond. And so can we, instead of getting caught up in all the horribleness of it, can we just put that aside and say, okay, there's nothing I can do about it, or, it's happening, what can I do, as opposed to go spinning in off on to wherever about it. This is a quote from a Will Smith movie. He says, "Do not misunderstand me. Danger is real, but fear is a choice." Again, sort of like pain and suffering. So we cannot control what is happening to us, but we can control how we respond to it. And in a certain life, Zen is feeling life instead of feeling something about life. And finally, this one little quote, which I love, from Joseph Goldstein, which is appropriate for right now. So, anything can happen anytime. We can be going along just fine in our lives, and suddenly there's an accident or an illness or some dramatic change in the conditions of the world. COVID. Some people may hear anything can happen anytime and think, oh my God, that's depressing. But rightly understood, it's not depressing at all. It's really freeing because it's understanding this. We are not living in, it's really freeing because in understanding this, we are not living in delusion. The mind relaxes, lets go of fear, and is much more open because we acknowledge the truth of change rather than deny it. And that's really what this is all about, right? Is we're not living in delusion. This is what's happening, right? COVID is happening. Everything that happened in the Capitol last week is happening. The last 4 years of Trump happened. You know, we can spin out with fear about it, or we can figure out how to respond in an adaptive way. And so with that, I think this is it. So thank you very much. And we can do some questions. And I'm gonna, go back to this. So, and I'm gonna stop sharing.